Good afternoon, I'm Kayla Baldwin, and today I'll be presenting my Week 4 case studies. Thank you.
For the first case study, we are presented with an image of an individual with a large tumor on the left hemisphere of the brain. On the left, an MRI image shows a gray and white scale of a tumor, and on the right, there is a PT scan of the tumor after receiving a dosage of fluorescent D-glucose. Based off of the visual differences, we can see that on both images, the tumor cells are brighter in contrast to the normal healthy cells. To find why that is, I'm going to explain the reuptake of glucose in different cells in the body and how it differs in tumor cells. Carcinogenic tumor cells or cancer cells are easily identifiable compared to normal cells from their growth rate, their shape, and abnormal number of chromosomes arranged in a disorganized fashion. Under normal circumstances where cells are perfectly normal and healthy, glucose can successfully be regulated in different tissues within the body. Glucose is an important factor in most of our biological systems for efficient energy production and cellular function. Our current image shows us an example of how glucose enters the bloodstream through facilitated diffusion in the skeletal muscle. The insulin hormone that is released binds to a receptor on the surface of the cell, which then triggers the GLUT4 proteins to be released from intracellular storage vessels within the cell. Glucose regulation in liver cells are depicted in the following figure. Hormones such as insulin and glucagon is released when glucose levels are either too high or too low. In the following diagram, we have the uptake of glucose in brain cells. Glucose molecules transport from the bloodstream by specialized proteins called GLUT1 or GLUT1. The glucose molecules then undergo glycolysis to produce ATP, where it then provides energy to important processes in the brain, such as synthesizing neurotransmitters, conducting membrane potentials, and signal transmitting. When our glucose levels begin to rise, the hormone insulin is released by the pancreas into the blood. Insulin will then bind to the receptors on adipose tissue cells that will trigger several responses. The glucose transporter protein GLUT4 travels to vesicles in the cell membrane and permits glucose to enter the adipose cells. Here we have a figure of two cells, which one's normal and one's a cancer cell. This figure shows us on the left the normal cell undergoing glycolysis, and on the right is the cancer or a tumor cell. The difference here is that the cancer cell is undergoing aerobic glycolysis. This is known as the Warburg effect. Research conducted by Kim S.H. and Baek K.H. on how the Warburg effect works. In comparison, the cancer cell has an increased rate of glucose uptake. This results in the cancer cell being able to survive, grow, and metastasize at a faster rate compared to a normal cell. The modifications that occur to alter glucose uptake in cancer cells includes expressing the GLUT1 and GLUT4 transporters at an increased rate, meaning glucose will have a higher chance at spreading to other areas of the body. Moving on to the next case, we are presented with a 16-year-old female that is unable to produce adequate amounts of muscle glycogen phosphorylase. When an individual has a muscle glycogen deficiency, it is diagnosed as McArdle disease, resulting in having a deficiency in the glycogen phosphorylase enzyme. This enzyme breaks down glucose into glucose 1-phosphate, which aids in muscle contraction. Symptoms that prevail in individuals with this disease can experience pain and muscle cramps even with mid-intensity movements, such as going on long walks. Individuals with McArdle disease do not have to worry about having reduced muscle glycogen stores because this disease does not mean that there are few numbers of glycogen stores. It particularly affects being able to use glycogen that is stored in the muscles. Another positive is that she would not have to worry about checking her glucose levels like one would with diabetes. If the body is at rest, or at the very least doing low activity, glucose levels should be within normal ranges. However, intense activity, she might experience muscle cramps and fatigue due to the excess lactate accumulation.